Welcome back to the online class, students. So in the previous class, we discussed about the introduction of enzymes. Okay, and so the definition of enzyme is it is a biocatalyst, which increases the rate of reaction. So as the enzyme acts as a catalyst by increasing the rate of reaction. So in today's class, we are going to discuss about how the enzyme is causing or responsible for high rate of chemical conversions. Okay. So in today's topic, we are going to discuss how the enzymes are responsible for bringing out high rate of chemical conversions. Okay. So in the previous class, we have learnt about some of the definitions related to the active site. Like active site. What is the active site? So active site is nothing but it is the pockets or crevices on the surface of the enzyme. Okay. So active sites can be defined as what? It is the pocket or crevices, means cracks. Crevices on the surface of enzymes. Okay, on the surface of enzymes. So which is responsible for catalytic activity of the enzyme. Okay, so the active sites are responsible for the catalytic activity of the enzymes. The next is a substrate. What is a substrate? Okay, substrate is nothing but it is a chemical. Substrate is a chemical, so which get converted into a product. Okay, substrate can be defined as what? It is a chemical which get converted into product. Which get converted into what? Product. Okay. Then next, otherwise even you can define substrate as it is a chemical on which the enzyme act. Okay. The next is the enzyme. So what is the enzyme? So as we know that enzymes are the catalyst. Okay. So as they are catalyst and even they are made up of what? Proteins. So enzymes are made up of what? Proteins. Enzymes are made up of proteins and they have three-dimensional structure. So they have what? 3D structure or three-dimensional structure. Okay. That three-dimensional structure even includes the active site. Three-dimensional structure also includes what? Active site. So the enzymes are proteins which are having the three-dimensional structure and are capable of converting or transforming the substrate. Transforming the substrate into a product. Okay. So this can be represented as substrate, yes, arrow, product. Okay. So now, as we know that the substrate bind to the enzyme in the active site region. Okay. So for which? First, the substrate has to move towards the active site. Okay. The first thing is, so the substrate has to move towards the active site of the enzyme. Active site of the enzyme. Later, so the substrate combined to the active site of the enzyme and results in the formation of enzyme substrate complex. Okay. So results in the formation of what? Enzyme substrate complex. Enzyme substrate complex. So this is an obligatory formation or obligatory complex formation. Obligatory means, so it is compulsory. If the substrate has to be converted into a product, it is compulsorily, it is supposed to bind with the enzyme and enzyme substrate complexes required in order to get converted into a product. And so this enzyme substrate complex is a temporary state or it is a transition state. Okay. So this is a short-lived unstable structure. Okay. So enzyme substrate complex is a temporary, short-lived and temporary structure. Okay. So it is a short-lived, a temporary structure and highly reactive. Okay. So this temporary formation of the short-lived complex formation, so such a phenomenon is called as transient phenomenon. Okay. So the enzyme substrate complex is a short-lived. So formation of such complex, we are calling it as what? Transient phenomenon. So it is called as what? Transient phenomenon. Okay. The next. So when this substrate bind to the active site of the enzyme, okay, so it undergoes some structural changes. Okay. So when the substrate combine to the active site of the enzyme, so it undergoes some structural changes. That means substrate gets a new structure by binding to the active site of the enzyme. It gets what? New structure. So this new structure is called as transition state structure. So it is called as what? Transition state structure. Transition state structure. The next. So now the transition state structure, that means whenever a substrate, a stable substrate has to get converted into a product. Okay. So it has to pass through transition state structure. Okay. So the conversion of this, the substrate into a product, it is a continuous process where the number of reactions will be occurring. So when a linked number of reaction occurs, that we call it as what? Pathway. So the pathway in which the transformation of substrate into product, so it has to go through what? Transition state structure. So the transition state structure may be many. So that means what? When a substrate gets converted into product, so in between, so it will have many transition state structures. Okay. Then let us know about the activational energy. Okay. So let us know about what? Activational energy. So what is the activational energy? So the reaction. So if a reaction has to occur, so it requires the external energy supply. Okay. So if a reaction has to start, it requires the external supply of energy. So to start the reaction, such type of energy is called as what? Activational energy. So we can define activational energy as a reaction to start, okay, external supply of energy is needed. So such type of external energy we are calling it as activational energy. So now, so this activational energy, so when an enzyme is present, so the enzyme reduces the activational energy required for a reaction. Okay. So what happens here? Enzyme reduces the activational energy required for a reaction. Okay. So this can be well studied by taking an example of a graph. Okay. So this can be well studied by taking an example of a graph. So here in the graph we'll be having x-axis and y-axis. Now on the x-axis I'm going to take the progression reaction and y-axis I'm going to take the potential energy. Okay. So this is the graph here. The graph represents x-axis and y-axis. So x-axis represents progression or progress of a reaction. Y-axis represents the potential energy. Okay. So this y-axis represents the potential energy. Y-axis represents the potential energy. So what is the potential energy? Potential energy is nothing but the stored energy. Okay. So that we are taking it on the y-axis, and on the x-axis we are taking what? Progression or progress of a reaction. Okay. So now let me write the graph. So I'm going to write the two graphs here. So one is in the presence of enzyme, one is in the absence of enzyme. Okay. So now first let me write in the absence of enzyme. Okay. So in the absence of enzyme. So this is a graph. So this point represents the substrate and this represents the product. Okay. So and in between, so the peak. So the peak represents, this peak represents the transition state. The peak represents the transition state. Okay. And so here you can see that, so this is the activational energy required for the initiation, for the activation of a reaction. Okay, so this is the external supply of energy which we require for conversion of a substrate into a product. So this is the activational energy. So here the enzyme is not the activational energy in the absence of enzyme. Okay, so let me write one more graph. So where the reaction occurs in the presence of the enzyme. Okay, so now, so this is the graph showing the presence of the enzyme. Okay, so here can you see here? So this is a reaction. So which is occurring where? So in the presence of enzyme. That means so only this much of activational energy is required for the conversion of a substrate into a product. Okay, so this is the graph. So showing what? Activational energy required, or activational energy in the presence of enzyme. Is the second graph. So here from the comparing the two graphs, you can conclude that so when the enzyme is there, the amount of activational energy required has been less. So when the enzyme is not there, the amount of activational energy required is more. Okay, so this is what the graph represents. Is it clear, students? So now, so with the help of this graph, so you can see that whether the reaction is exothermic or endothermic. So now just you see the energy levels of the substrate and the product. So the energy level of the substrate, so here it is more when compared to the product. Okay, so when the energy level of the substrate is more when compared to the product, such type of reaction we'll call it as exothermic reaction. So what we'll call? It is called as what? Exothermic reaction. So where? So there is a release of heat or energy takes place. Heat is released. Okay. So the next, if a graph in which, so the energy level. So here what we have is the graph showing the exothermic reaction. If any graph which shows the energy level of substrate is lesser than the product, that means, so here the heat or energy has been absorbed. Such type of reaction we will call it as endothermic reaction. It is called as what? Endothermic reaction. So here heat is absorbed. Heat is required or absorbed. Okay. The next, even activation energy we can define it as here. So the amount of energy required for conversion of a substrate into a transition state. Okay. So the amount of energy required for converting or for transforming the substrate into a transition state, that is the activational energy. Okay. Now let us study the mechanism of enzymatic action. Okay. So now let us study the mechanism of enzymatic action. So here actually there are two types of mechanisms. Okay. So there are two models which will be explaining about the nature of enzymatic action. Okay. So which is there in our syllabus. It is lock and key model. First we'll study that. Okay. So nature of enzymatic action. So the next subtopic is nature of enzymatic action or nature of enzymatic mechanism. Okay. So now, as we know that, so the enzyme combines with the substrate. Our substrate combines with the active site of the enzyme. Okay. First, let me write that. So, enzyme is the substrate combines with the active site of the enzyme, results in the formation of a temporary state called as what? Enzyme substrate complex. Okay. Later, this enzyme substrate complex So get converted into enzyme product complex. It get converted into what? Enzyme product complex. Enzyme product complex. Now later they will get dissociated into enzyme and product. Okay, so let us study this one with the help of a diagram. Now, as you know that enzyme consists of the active site. Okay, see for example, so this is the enzyme here. Okay, so it is having the active site. So it is having the active site. This is the active site. This is the enzyme. This is the enzyme. Now, what happens to the active site which will get combined there? The substrate will get combined. Okay. So, for example, so this is the substrate. So, this is what? Substrate. Okay. So, when substrate combined to the active site of the enzyme, it results in the formation of what? Enzyme substrate complex. Results in the formation of enzyme substrate complex. Okay. So, this is the enzyme substrate complex which is formed here. Enzyme substrate complex. Later. So, these enzymes are nothing but which helps in the breakdown and formation of the chemical bonds. Okay. So, here what happens? So, the in the substrate, so the breakdown or formation of chemical bonds takes place with the help of the enzyme. So, when that breakdown or formation of chemical bond occurs, the substrate will get converted into a product. Okay. So, now it results in the formation of enzyme product complex. Okay, enzyme product complex. Now, what happens? So, the product gets separated or product gets released from the enzyme. Okay. So, the products gets released from the enzyme. For example, so you can take this as the product. Okay, so this is structure of the product. It gets released from the enzyme. Now, again the enzyme is free. So, this free enzyme can again get combined to the substrate and later the process of conversion of product will be continued. So, here you should remember, so for each active site, for a particular active site, a particular substrate can bind. Okay, so this model is called as lock and key model. Okay, so enzyme act as a lock and the substrate fits like a key. So this is a lock and key model. So which we have discussed now is the lock and key model. Okay, so this lock and key model was proposed by Fisher. That is Emil Fisher. Emil Fisher proposed this lock and key model of the enzymatic action. So another theory is there, or another model for the explaining the enzymatic action. So that is induced fit theory. So that is what? Induced fit theory. What is the meaning of induced fit theory? So in case of induced fit theory, this is proposed by a scientist by name Coastland. Okay, it is proposed by a scientist by name Coastland. According to him, when a substrate bind to the active site, okay, when a substrate bind to the active site of the enzyme, so according to Coastland, so the active site undergoes structural changes so that it can firmly bind to the substrate. Okay, before the active site undergoes changes, the distance between the substrate and the active site was more. So when the active site undergoes structural changes, so the substrate and active sites come together. So it helps in the bond formation. Okay. So according to the induced fit theory, active site undergoes structural changes so that it can firmly bind to the substrate. Okay. But here, according to the lock and key model, active site does not undergo any changes. Okay. Is it clear, students? So this is regarding the enzymatic action. So as you know that, the enzyme act as a catalyst. So if the enzyme should act as a catalyst, so the obligatory complex formation, that is enzyme substrate complex, is required. Okay. So let us see. So the catalytic properties. So the catalytic properties of the enzyme. Okay, so the first you know that the enzyme combined to the active site. Sorry, the substrate combined to the active site. To know the catalytic property, okay, so it has been described as follows. So first thing is, what happens here? Substrate bind to the active site of the enzyme. Substrate bind to the active site of the enzyme. Okay. Then later what happens here? Now the enzyme substrate complex has been formed. Next step is enzyme substrate complex. So when this enzyme substrate complex has been formed, so the active site of the enzyme undergoes some structural changes so that enzyme can hold the substrate firmly. Okay, so the enzyme undergoes some structural change. So due to the structural change, so the enzyme holds the substrate tightly and it helps in the bond formation and breakdown. So later, after the breakdown and bond formation, enzyme substrate complex has been converted or transformed into a enzyme product complex. Okay, so as enzyme product complex is formed, so next step what happens? Enzyme releases the product. So enzyme releases the product. Again the enzyme is free. So that means a catalyst means it should not undergo any structural changes, isn't it? So it should not undergo any structural changes. Again, the enzyme can bind to the substrate and it can convert the product. So the process continues. So this is regarding the today's class, and let us see some of the questions related to the topic. Okay. So the first question is, transition state structure of the substrate, transition state, temporary state of the substrate is formed by enzyme reaction is a permanent but unstable. Second one, transient and unstable, permanent and stable. Fourth one, transient but stable. So as I told you, it is a transient, temporary, and it is an unstable structure. The answer is B. Second one, an enzyme brings about decrease in reaction time, increase in reaction time, increase in activation energy, decrease in activation energy. So enzyme, always in the graph we saw that enzyme reduces the activational energy. Answer is D. So enzyme has dash structure. Okay. So primary, secondary, tertiary, quaternary. Okay. So enzymes are made up of proteins and they have the tertiary structure. Answer is C. Then the fourth question. So external supply of energy is required to start the reaction. It is called as activational energy, potential energy, kinetic energy, none of this. Okay. So the external supply of energy, we call it as what? Activational energy. The answer is C. So this is related to today's class, students. Thank you.